Good afternoon. I'd like to thank the Congress organizers of HIV-NAT and you, the audience, for joining me to talk today about broadening the blood donor base, inclusion of LGBT populations in blood donations. My name is Brian Custer. I'm an epidemiologist. I work at Vitalant Research Institute in San Francisco, California, and hold a joint appointment in the Department of Laboratory Medicine at UCSF. I do want to cover my outline for the topics I'll be sharing with you today. I want to provide a conceptual framework. Many of you may not be familiar with blood donation or blood donation rules, so I want to give you at least a small background to cover that helps then define what I'll talk about specifically with respect to LGBT donors. I do want to discuss changing approaches to donor deferral, the use of shorter deferral periods, and current considerations and possible concerns. And finally, I will, toward the end of the talk, discuss new directions, actually, quite honestly, exciting new directions in how decisions are being made with respect to expanding the donor base to LGBT donors. So to begin with, I want to give a brief overview of what is called the Blood Donor History Questionnaire or interview. This is a series of questions about a donor's potential health exposures and behavioral history. These questions are asked to understand if there's a risk to the donor in donating and if there's a potential risk to recipients who might receive the donor's blood. Because there are specific risks to recipients associated with history of pregnancy, females are asked female-specific questions. Because there are specific risks to recipients associated with sexual histories, different questions are asked of males and females. These questions are primarily focused on whether you are a man who has sex with other men or a sex partner of a man who has sex with other men. But it's actually for these reasons that there are different questions that males and females are asked in most contexts related to blood donation. This creates complexity in expanding the donor base. I'm also going to give this through the background of the US FDA policy. It's obviously only one example, one country that has a series of policies, but that policy on particularly men who have sex with men has really changed quite dramatically over time. In 1983 through 1985, men who have sex with men or gay and bisexual men were asked not to donate blood along with other groups that were identified as higher risk for HIV infection at the time. Starting though in 1985, this changed to be a permanent deferral or indefinite deferral for any man who had sex with other men and reported that having occurred anytime after 1970. And so from that period of 1985 to January of 2015, any man who had sex with men was indefinitely deferred if they disclosed that at the time of donation. However, really, these policies have started to change. They've started to change globally. In the US, in the period from 2015 to April of 2020, there was a one-year deferral since last MSM sex. More recently, as early as April of 2020 this year, that changed from a 3-month deferral since last sex with another man. All of these successively lower time periods for deferral are linked with our ability to detect and identify HIV infection in a donated unit of blood. This topic, while you might not be familiar with it, is actually not new. It goes all the way back to 1968, or over 50 years now, a steadily increasing body of published literature on this topic. And so again, this graphic is just intended to give you an overview that there has been research done going all the way back into the 60s, but as you would expect, not until really the formal identification of HIV in the early 80s, and then the period thereafter where we started to see progressive changes in policy, have the number of publications increased dramatically over time. So I do want to describe what may be evident to some of you, but is very important. Really, different sexual and gender identities lead to different rules for blood donors. For somebody who is a lesbian or a female who has sex with other females, generally the deferrals or the eligibility to donate is the same as it would be for heterosexual donors, unless the same sex behaviors are not accepted in the broader society. So of course it is context or country specific. For gay and bisexual men, the focus is on HIV risk, and generally it's a behavior-based deferral in most settings, meaning if people are highly sexually active, so have many sexual partners or have recent exposures, they may not be allowed to donate. For bisexual women, it's different. It really would do the same as it is for lesbians or heterosexual women, unless they happen to be a sexual partner of a man who has sex with men, in which case they would actually have MSM-style questions asked of them. And for transgender individuals, this is the most complex topic where really there's quite a bit of variability in rules. Really, we don't have a strong evidence base at this time. So for the rest of this talk, I'm really going to focus just on gay and bisexual men and also cover a little bit about the current status with trans persons and their eligibility to donate. Give a little more background, there really is no consensus on international policy at this time. There's factors that influence policy, and these are different in different contexts. So really, there may be 6 main things to be cognizant of. One is the epidemiology of HIV and what it is in the specific country or local setting. Clearly, if it's a primarily heterosexually spread epidemic, then there will be different rules than if it's a primarily homosexually spread or spread by men who have sex with men. So that epidemiology of HIV is very important. In addition to that are the actual blood donor screening methods that are available and in use in any given jurisdiction. Most developed countries screen both for nucleic acids, for direct detection of the virome of HIV, and also for antibodies, so antibody screening. So these dual strategies allow us to really be confident that we've identified HIV if it is present in a blood donation. Local regulatory requirements, so the policies such as the FDA makes drive what will be allowed or not. But in addition, there are political or government decree aspects to this. So policy can change if governments change or if legal rules change in a given context. Risk analysis and modeling to project what the risk might be under different policy scenarios has been used successfully to really understand how might policy impact risk. And then the history of response to threats to blood safety is a critical aspect of how a given jurisdiction may handle blood safety policy. This is because really the tragic history of transfusion-transmitted HIV and other infections, hepatitis C in particular, during the period of the 80s and the 90s. So broader contextual considerations are also important to keep in mind. There is a high level of controversy around what constitutes evidence or science-based policy in this domain, and it makes it very difficult for everybody to reach consensus understanding. The current testing approaches, however, really in developed countries do not justify an indefinite deferral for any LGBT donors. And the reason for this is very clear. The primary concern continues to be HIV infection and other known infections. If you were thinking about emerging infectious diseases as a primary reason to not change policy, that's really not justified by the patterns of emerging infectious diseases that we've seen over the last 20 years in terms of infections that are relevant to blood safety. Those infections have primarily been vector-borne infections, and so they really are not going to be human-to-human spread infections. It is true that analyses of changes in donor deferral periods for MSM show that gains to the quantity of blood in the supply are marginal, meaning even if policies become very open and allow gay and bisexual men and trans donors to donate, this isn't going to lead to a huge increase in the number of donations coming into the supply because these members tend to be a relatively small proportion of any population. But there are some bigger consequences. It really is the issues and the implications related to how, as societies, we view equity that really do impact this and have a negative impact that's larger than you might expect, because if policies for blood donation are not considered equitable for everybody, then this may have a negative impact even on people who are otherwise eligible to donate. And so it has this overall impact of reducing the willingness of people to participate in donating blood. So the next couple of slides are just going to speak to what is the current status related to transgender or trans donors. This is a similar graph to the one I showed with respect to men who have sex with men, but you can see it doesn't start in 1968, it starts in actually 2014. So published studies thinking about and commenting on transgenders, population and blood donation is a new phenomenon, and very few studies have been published. So the data are limited, and there definitely is more variability in policies than for gay or bisexual male donors. What really is known is summarized very nicely in a recent publication from Canada. There are different approaches that can be used for selecting to determine if a donor is eligible if they're trans. One of them is a medical inquiry process, or basically individual risk assessment. So using a guided conversation with a medical professional to determine if a person has risks or not. Another approach would be the donor history questionnaire, but the donor history questionnaire is not well suited for trans donors, particularly, as I said, because you may want to ask questions that are applicable to both males and females, and that's a difficult thing in this context. So one approach might be to ask persons to complete both male and female questions on the donor history questionnaire. However, this approach has not been studied, so we don't really know if it's considered acceptable or not to trans donors. Another approach might be to ask questions on your sex assigned at birth and then gender-specific questions. This approach also might be viable, but again, it hasn't really been studied for its acceptability to trans donors. So where does this leave us? Really, the greatest impediment to facilitating blood donation in a way that is fair and safe for trans donors and recipients of blood is the lack of relevant and specific research. We need more studies on the epidemiologic study of sexual risk factors in trans persons, and we need to assess optimal approaches to donor screening questions specific to gender diversity. These studies are starting to be done at this time, but the data really have not yet impacted policy in a substantive way. So the rest of the talk is really going to focus on how the policy for men who have sex with men is changing in different contexts. And to do that, really, there are different ways of thinking about how you would defer or not an individual who might have a risk behavior related to being a sexually active men who have sex with men. You can have a time-based deferral. That might be an indefinite deferral, like I said earlier on. It could be a 5-year deferral, a 1-year deferral, or a 3 or 4 month deferral, any specific amount of time it can be. So in this case, it could be if you reported that you had sex with another man at within that time period, whatever it is. And if you did, then you'd be given a deferral for up to that time period as a way of reducing the risk of HIV infection. Another approach for eligibility assessment would be to ask about specific activities. These are gender neutral policies and certainly preferred from a broader perspective. So regardless of whether somebody defines themselves as gay or bisexual, you might ask questions, or heterosexual, you might ask questions about sexual behaviors that are considered high risk, regardless of whether the partner is the same or opposite sex. And these could be in a specified time period preceding the donation, and then a deferral would be given, but it would be applied to everybody. So that's why it's called gender neutral policies. A similar approach is the individual risk assessment. Again, that structured dialogue with a medical professional. This is maybe the most equitable way of selecting donors, but it actually is quite complex and is not used in many settings because of how expensive it is and the time required to appropriately understand the individual risks that a person may have. And then another approach for eligibility is to have alternate policies. So these are unique policies that permit men who have sex with men or transgender donors to donate either a specific type of blood component, in this case plasma, or potentially platelets. And then those are actually treated in a manner that we're confident that there's a low risk of infection in those. And so this is one possible way of expanding the eligibility for LGBT donors. So I do want to give you a appreciation for how complex this topic is by briefly reviewing the variability in how policies are established, particularly for sex between men. In the countries listed here on the left, you can see that there's no specific risk that is defined unless it's a high risk. So this is countries that don't have a risk that has a time-based deferral for men who have sex with men. And instead it's based on if some type of risk is reported through that individual discussion that puts them at risk. So countries as variable as Argentina, Hungary, Spain, South Africa, and Brazil have some form of this. Now, how exactly it's implemented and how the rules are applied is going to be as variable as the countries themselves. By far, the most common approach is some form of these time-limited deferrals. And so, as you can see, currently in the UK, it's a 3-month deferral. In Canada, it's a 3-month deferral. In the USA, it's a 3-month deferral. In France, Denmark, and Netherlands, it's currently a 4-month deferral. Japan has a 6-month deferral since last sex. Australia has a 12-month deferral, although that is expected to change to 3 months very soon. And Germany currently has a 12-month deferral. So again, these are policies that are time limited in the 3 to 12-month range. But there's still other countries that maintain a permanent deferral or an indefinite deferral for men who have sex with men. That includes Austria, although that's under review at this time, and other countries such as Belgium, China, Croatia. So the rest of the talk is really going to talk about how the policy has changed and the kinds of data sources that have been used to implement policy change in different contexts. I'll begin with the United States, the country that I know the best. I'm a part of one of the investigators that participates in what's called the TIMS program, or the Transfusion Transmissible Infections Monitoring Program. In that program, we are collecting blood donor data from approximately 60% of the US blood supply. That's about $6 to $7 million, 6 to 7 million donations per year represented in the databases. We use that to actually track prevalence and incidence in donors. We also use this structure to do other studies, and I'm going to mention two of those. One is related to HIV testing and antiretroviral use in blood donors, and then also studies of PrEP testing or PrEP in blood donors. So when the policy changed from an indefinite deferral to a 12-month deferral for men who have sex with men, we needed some time to pass for us to then be able to assess, has the incidence of HIV in the blood supply, in the donors who are donating the blood supply, changed in any way since the implementation of that 12-month deferral? And so we recently reported results comparing that period from 2015 to 2016 of an indefinite deferral to the period from 2016 to 2018 where the 12-month deferral was in place in the United States. And what we saw when we look at either female and male donors or male donors alone as first-time donors, we saw no statistical evidence that there was an increase in incidence when the policy was reduced to the 12-month deferral. That actually translates into no evidence of an increase in the residual risk that HIV would be in the blood supply in the United States after the change to the 12-month deferral. So that's very reassuring. And I do have to say that all of the studies that have been reported for countries like Canada, for France, for the United Kingdom, when those policies have been reduced in terms of the deferral has been reduced in time, we have not seen an increase in incidence. So nothing that suggests that going from indefinite to 12 months to 3 months has so far shown an increase in risk of HIV to recipients of blood. But we have a changing scenario now, and that is related to the much wider spread use and availability of antiretroviral therapies, of course, to treat HIV, an incredible advancement in our ability to control the epidemic, and even more so, the availability of PrEP to prevent HIV acquisition altogether. These have potential implications for our ability to detect HIV in the blood supply, which is why we're concerned about them. So pharmaceutical success in HIV control and prevention may not directly translate to blood safety. So there are a couple of commentaries that have been published thinking about this particular topic, but it can be summarized in this way. There is a potential conflicting message between undetectable equals untransmittable in public health or for sexual contact, where if somebody has an undetectable HIV infection, to the best of our understanding, it's untransmittable. That may not be the circumstance for blood components and blood units. And that's just simply because the amount of blood exposure, the amount of blood actually transfused intravenously is so much greater that even if a very small viral load is present, you still potentially could have a transfusion-transmitted HIV infection. So we're concerned, and we need to understand how much antiretroviral therapy use and also PrEP use may impact our ability to detect HIV infection in blood donors. So this is also linked to the testing that we do. As I said, typically most countries do nucleic acid testing for RNA and also antibody screening. And so these are data from South Africa and exhibiting what seemed to be a very disturbing or difficult to understand trend, which is elite controllers, so people who are antibody positive but RNA negative, seem to be increasing over time. But actually, ultimately, this was not the case. The explanation was that donors were coming in to donate, and they were taking antiretroviral therapy and donating blood. So this is a surprise and unexpected, because if you're HIV positive or if you're taking antiretroviral therapy, these are the kinds of questions that you're asked on the donor health history questionnaire. So this phenomenon is stating that there are clearly individuals donating who are not disclosing HIV infection or antiretroviral therapy. We asked a similar question in the US, would we see that same pattern? And the answer is we did see evidence that people who are HIV positive are coming in to donate, and they are on antiretroviral therapy at that time. So we did a much smaller study, but in that study of around 299 HIV positive donors, when tested for antiretroviral drugs using pretty sensitive, highly quantitative assays, 15% of those HIV positive donors had evidence of antiretroviral drugs in their system at the time of donation. Comparison group of HIV negative donors had no antiretrovirals in their system. We then specifically looked at first-time male donors who came in to donate but were infection negative. And it's, again, a relatively small study, only 1,500 individuals. But of those 1,500 individuals, 0.6%, or 9 individuals in total, had PrEP in their systems at the time that they were donating blood. That's actually a good thing. That means that there's low risk of HIV infection, but what we're worried about is a potential PrEP breakthrough infection in the donor population. So I'm going to touch on really, I think, the most interesting and dramatic country in the sense of making really major inroads into trying to increase the eligibility and the opportunity for donors to donate. So currently in the UK, there is a 3-month deferral. That 3-month deferral has been in place since 2017 in England, Scotland, and Wales, and since June of 2020 in Northern Ireland. That policy of the 3-month deferral since last sexual contact for men who have sex with men also applied to commercial sex workers and sex partners of groups known to have a high risk of having a sexually transmissible infection, so any kind of high-risk partner. And all of those individuals were treated the same way in that it was a 3-month deferral applied for any of those potential risk exposures. But there's still been a lot of pressure in the UK, as in other countries, to make further changes to try to move away from a time-based deferral. And indeed, the UK, if you're aware of recent news from just December of last year, just indicated a major shift. So that shift is based on, for the assessment of individualized risk, or FAIR. FAIR was a series of studies that I will describe in just a moment, but I think that the punchline, what's happened, is actually the most important thing, so I'll give you that summary first. So just about a month ago now, the National Health Service removes the 3-month barrier for men who have sex with men and others, and says that gay and bisexual men who have had the same sexual encounter for more than 3 months will be allowed to donate if there's no known exposure to a sexually transmitted infection and they are not using drugs to stop the spread of HIV, the virus that causes AIDS. These new rules are going to apply across England, Wales, Scotland, and Northern Ireland. So FAIR, for the assessment of individualized risk, is really based on a series of studies that started in February 2019, culminating in a final report issued in October of 2020, in which really studies of the epidemiology of infections in donors, studies of behavior, and studies of psychosocial impacts of different approaches to asking questions on the blood donor questionnaire were analyzed and assessed for a path forward to be able to implement those. Now, it does say, as you can see, in March of 2020, COVID-19 certainly shifted the timeline a little bit. And so FAIR, this policy change, has been adopted, but it won't really be implemented until sometime in 2021 now. It's based on a large body of work to try to understand, again, bloodborne infections, donor behaviors based on survey, and thinking about both through focus groups and retest studies, what questions might be appropriate to ask donors. So how will this actually work? It's planned to implement in mid-2021, as I indicated. Under the new changes, all donors who have ever had sex will soon be asked to also consider their recent sexual activities before giving blood, and this will be on the donor history questionnaire. The donor history questionnaire will be a cascading or hierarchical set of questions. So to really understand risk, they're going to ask increasingly detailed questions, and a combination of those are then going to be used to determine whether a man who has sex with men is eligible to donate or not. Deferrals will still be made for individuals if they report any of the following. Anyone who has had anal sex in the last 3 months with a new or multiple partners, regardless of whether they report condom use, will not be allowed to donate. They'll have the 3-month deferral. Anyone who has reported chemsex, so having sex under the influence of illicit drugs in the last 3 months, will also not be allowed to donate. If an individual has received recent treatment for gonorrhea they will not be allowed to donate, or if they are receive treatment for syphilis, they also will maintain that they'll not be allowed to donate. Linked to the concern about PrEP, a 3-month deferral for anybody who is taking PrEP will remain in place, although they are currently reviewing their PrEP guidelines to try to understand if that could also be modified. And the screening of donors for syphilis, even though syphilis is not considered a transfusion transmissible infection of high concern because of the treatment procedures that we use, it still will be maintained as a useful safety measure to monitor donors for evidence of increased sexual risk behaviors. I'm going to shift just briefly to say, so that's the way that the UK approached making modifications and really expanding the donor base as much as possible with that structured approach. Different jurisdictions have different considerations. Here in Brazil, the Supreme Court made a decision. The court ruled that all blood collectors in the country must change the pre-donation interview, including not allowing blood centers to ask blood donors whether their sexual partners are the same or opposite sex, or inquiring about other aspects of the donor's sexual partners. So it's a very different process, but it ultimately leads to the same result, which is having to modify to create a much more inclusive approach for blood donation. So blood centers are still in the country of Brazil are still working through how to implement appropriate questions. Most will rely on the number of sexual partners as what they use to define the risks. Quarantine plasma for men who have sex with men has been a policy that's been in place since 2016 in the country of France. So this is actually a situation where all donors are treated exactly the same, whether they're heterosexual or homosexual. And it's based on the approaches that the overall system uses to have a quarantine system for plasma. This is where somebody donates a unit of plasma, and that plasma is held until the individual returns a few months later and either donates again or provides a sample for testing. That test sample is then tested, and if it tests negative for all of the infections, then that plasma that was previously collected can be released for transfusion. So they just allow men who have sex with men, if they've only had one sexual partner in the last 4 months, to participate in that same program in exactly the same way as applied to heterosexual donors in the country. The last topic I want to touch on is a new study being done in the US that is really intended to provide information that may be similar and lead to a similar outcome to the FAIR initiative in the UK. And that's the ADVANCE study, or Assessing Donor Variability and New Concepts in Eligibility. So this is a study that's being conducted to provide FDA with evidence to which to consider potential changes to MSM donor deferral policy while maintaining the safety of the blood supply. The objective is to conduct a pilot assessment of the discriminant function of revised donor history questions for predicting recent infection with HIV in men who have sex with men who wish to donate blood. So again, the study that we'll conduct will actually assess questions. If those questions are shown to differentiate different risk groups among the men who have sex with men population, those will then be potential candidates for inclusion on the donor history questionnaire in the United States. So I do want to summarize my presentation today. Thank you very much for your time. But I will say that blood donation policy for men who have sex with men is evolving rapidly in several countries. And I would say now also donor policies related to trans donors are also evolving rapidly in several countries. Ever shorter time-limited deferrals are increasingly being adopted. The UK continues to be the leader and has announced a breakthrough. This cascade of behavior-based questions in a non-gendered manner is the really important breakthrough in that inclusion of a broad group of donors in the donor base. Other countries have or are considering special programs to allow MSM to donate specific components. The local epidemiology and public health considerations are even more relevant now. You really have to understand the local epidemiology to be able to understand what kind of policy might be appropriate. But I do want to end with one note of caution. It really is the local studies have to be done. You cannot rely on data or information from another setting and apply that to your own setting. The work has to be done to understand the local epidemiology and what that might mean specifically for LGBT blood donors. I do just want to conclude by acknowledging all of the study participants and blood donors who have taken part in research to inform blood donation policy in different countries, all of the research and blood center operations staff who have conducted these studies, and then specifically name Mindy Goldman, Sheila O'Brien, Katie Davison, Pierre de Bergen, and also Tassila Solomon, who provided specific information from the countries that I presented information on today. I also would like to thank you again for listening. And if you're interested in speaking to me or following up with me, you can see my email address here at the bottom of the page. It is bcuster@vitalant.org. I'd be more than happy to talk to you about the issue of expanding the donor base to LGBT population. Thank you again.